Great. So I think it's time we start and more people can join later on. So Happy New Year, everyone, and welcome back to our virtual journal club series. And before I start and introduce our lovely speakers for today, I would like to remind you of our ground rules as we do every time. So please make sure you keep your microphones muted during the talks and raise your questions by using the chat button or raising your virtual hand at the end of the talk, preferably. Finally, we would like to thank Dr. Vatakis and Dr. Weiner, as well as the ACR committee for putting these meetings in place, and of course, all of you for attending every time. So our first speaker is Francesca Barbero, who is a postdoctoral research fellow at the Rhythm and Brains Lab in Belgium. Her PhD was on voice perception and experience-dependent plasticity associated with the processing of socially relevant sounds. Her main research interests include understanding how humans make sense of what they hear and how auditory perception is influenced by different sensory motor experience and multisensory contexts throughout the lifespan. So today she's going to talk about rhythm categorization in human brain activity. Francesca, the floor is yours.
Thank you. Thank you for the introduction and thank you for having me here today. So in my talk of today, I would like to present to you a new stream of research that we are developing in the Rhythm and Brains Lab of Professor Silvino Zaradan in order to reveal rhythm categorization in human brain activity. Oops. Okay. So music is present in every known society, and we tend to be engaged in musical interaction since very early in life. In fact, humans show a remarkable capacity to perceive, learn, and produce musical rhythms. And in the lab, we take musical rhythm processing as a unique way to understand brain functions. So in music, when we talk about rhythm, we talk about the timing of events of musical sounds that occur over time, or in other words, of the organization of the patterns of duration which are present in music. So if we were to take a rhythmic pattern, as the one that you can see here on the screen, we can first notice that it is made of 3 time intervals, where the first 2 intervals have the same duration, while the third one is twice as long as the first 2. And this is actually a rhythmic pattern that we can find at the start of a very famous song, which you will probably recognize when I play this rhythmic pattern to you. So here is just a little visual hint before I play the sound. So probably many of you were able to recognize this rhythmic pattern as the starting tune of Jingle Bells. Now, what is interesting for us here is that these notes do not necessarily need to be played at these exact same positions over time for you to recognize the Jingle Bells pattern. In fact, these notes could be played a little bit earlier or a little bit later in time, and you will still be able to recognize this pattern as the start of Jingle Bells. Now, interestingly, if we were to stretch the position of these notes over time until we get to another rhythmic pattern where all the 3 intervals have the same duration that you can see here at the bottom, and this would be something sounding like a metronome. Now, if I were to play all these gradually different rhythmic patterns to you, you will not perceive them as gradually changing over time with a gradual variation from jingle bells to a metronome. But rather, you would perceive the first patterns here at the top sounding like jingle bells until when there would be a switch in the perception and you would perceive the other patterns here at the bottom sounding like a metronome. And this is a phenomenon that is referred to as a rhythm categorization. So rhythm categorization has been demonstrated at the behavioral level using a wide variety of tasks. And it has also been tested across countries in the world, showing both commonalities and differences across cultures, with, on one side, some rhythm categories that seems to be universally present across cultures, while on the other side, some other rhythm categories seems to be culture-specific. However, notwithstanding the extensive behavioral work that has been described in the literature, what is still far from understood are the neural bases for this rhythm categorization. And this will be the focus of my talk today. And in particular, in the lab, we're interested in understanding how the brain encodes and processes patterns of time intervals in order to form internal rhythm categories. And to investigate this, together with my colleagues, we have developed a new method that is based on electroencephalographic recordings, frequency domain analysis, and the representational similarity analysis framework. So what did we do? We considered two interval rhythm patterns, as you can see here, and we considered a two-interval pattern that is characterized by a 1-to-1 ratio, meaning that it is composed by two intervals that have the same duration, and another pattern with a 2-to-1 ratio, where one interval is twice as long as the other one. And these are two integer ratios that are among those that are universally found in music across the world. So we took this rhythmic pattern and we looped them over time, so to have rhythmic sequences where these patterns were repeated. And then we created intermediate conditions with sequences in which the position of the second note was gradually shifted over time. So to have now a total of 13 conditions in this temporal continuum from a 1-to-1 to a 2-to-1 ratio. And then we presented these sequences to the participants while we recorded the neural activity using EEG, asking the participants to listen to the sequences without moving. And in a separate session, we also recorded the behavioral data by asking participants to tap their finger in synchrony with what they heard as a behavioral index of rhythm categorization. So after we collected this data, we first looked at the behavioral results, looking at the interval ratio of the responses. And so what I will plot here is the interval ratio of the tapping responses on the y-axis, plotted as a function of the interval ratio of the stimuli on the x-axis. And here, the dotted line represents an identity line, which means that whenever there will be a dot laying on this line, it means that there was a perfect reproduction of the stimulus while tapping. And what we can appreciate is that participants tend to distort the stimuli by producing a similar response to conditions that are close in time to the 2-to-1 pattern, until when there is a switch in the response, and then participants start to tap similarly across conditions all the way until the 1-to-1 pattern. And this is in line with a large number of previous studies that have showed this categorical bias in Western participants using similar stimuli. And so after this first sanity check, we then moved on to the main analysis of our study. And what we did is that we took the signals from the stimuli, from the EEG responses, and from the tapping responses, and we analyzed them using a novel approach that we will refer to as frequency RSA, as it combines frequency domain analysis and the representational similarity analysis framework. And we used this approach so that we could move from a unit-specific to a unit-free measure so that we could see whether and how the representational geometries of our different measures related to each other. So what did this approach consist of? So we had our 13 sequences represented over time here. So here what I'm showing is just one repetition for each rhythmic pattern for each condition. But remember that these patterns were looped in order to form long rhythmic sequences. And we extracted the envelope of these sequences by using an Hilbert transform. And then we looked at the data in the frequency domain by applying a Fourier transform so that we now have a frequency spectrum for each one of our condition, where we expect the stimulus to project only and specifically at frequencies that correspond to the duration of the rhythmic pattern and the harmonics. And we use these values then to build a representational similarity matrix by computing pairwise correlations between the frequency spectra across all conditions. And when we do that, we get a similarity matrix that is symmetrical around the diagonal. So here in this matrix, as in all the matrices that I will show you during the presentation, whenever we have a yellowish color, it means the two conditions were very similar, while instead when we have a more bluish color, it means that the two conditions were dissimilar. And what we can appreciate here is that as we move away from the 1-to-1 to the 2-to-1 condition, there is a gradual change as the spectra become progressively more dissimilar from each other. And this is expected from the way that we constructed our stimuli. And so we performed this procedure on the EEG and on the tapping data, with the only difference that we didn't use the amplitude FFT spectra that I showed you here for simplicity, but the complex spectra to keep both the real and the imaginary part of the spectrum. And so let's look at the results. So here is again the representational similarity matrix of the stimuli that I just showed before. And when we look at the representational similarity matrix of the tapping data, what we see is quite different. In fact, we don't see, as we saw for the stimuli, this gradual change from the 1-to-1 to the 2-to-1 condition, but instead we see a categorical structure in which responses that are close in time to a 1-to-1 ratio are very similar to one another, until there is a switch in the response and participants start to tap very similarly to rhythmic patterns that are close in time to a 2-to-1 ratio. And then when we look at the EEG results, and here is the topography of the response that we obtain when we average the data from all the conditions and all the participants together, where we see this clear frontocentral distribution of the response that we can typically observe in auditory experiment. And when we look at the similarity matrix of the response, we can again see this categorical structure that is remarkably similar to what we obtained for the tapping data, where responses to patterns that are close in time to a 1-to-1 ratio elicit very similar response, until when there is again a switch in perception along this temporal continuum, and responses to rhythmic patterns that are close in time to a 2-to-1 ratio are very similar to each other. And this actually constitutes, to our knowledge, the first neural evidence for perceptual categorization of rhythm. Now, to be able to assess whether we could have a statistically significant evidence in favor of a categorical representation of rhythm in the brain, what we did is that we built categorical models of perception that you can see here. And we built several of them by changing the location of the boundary for the switch between the two categories to happen, not to make any a priori assumption about that. And then we run partial correlations to that will be our statistical test. So I will show you here how we did it for the EEG data, but in fact we applied the same procedure for the tapping data. So we consider the individual data from each one of our participants, and here is the data from one of them. And we perform the partial correlation between the EEG data and all possible categorical models of perception by regressing out the information carried by the stimuli, which allows to control for potential acoustic confound. And from this, what we get is a winning categorical model, that is the categorical model that correlates the most with the individual EEG data, regressing out the stimuli, and we store this correlation coefficient. And we do this for all our participants, and then we can assess the significance at the individual and at the group level by running permutation tests. And what we observed is that for the EEG data, we got evidence in support of categorical representation of rhythmic patterns in the brain at the group level, and with results that were also significant at the individual level in most of our participants. And the same was also true for the tapping data. And then what you can see here on the right are the superimposed winning categorical model for each participant for the EEG and the tapping. And what we can appreciate from here is how the winning categorical models are pretty consistent, first across participants in the EEG and in the tapping responses, but also between the EEG and the tapping responses. So with this study, we extended the previous findings on rhythm categorizations, which had been limited to behavioral measures so far. In fact, using frequency domain and representational similarity analysis, we could show that the brain activity that is captured with EEG goes beyond the mere tracking of the physical properties of the stimuli and instead show categorical representation. We also showed that neural rhythm categories can emerge automatically in the absence of any timing-related explicit task and in the absence of movement. Yet, we could observe the representational geometries that we observed from the brain responses and the tapping responses were remarkably similar. So from this, based on these findings, we are now working on some future perspective using this novel approach. And in particular, during the last minutes of my presentation, I would like to show you some work that we are currently starting to work on in the lab to clarify the role of experience on rhythm categorization. So in a first study, the aim is to investigate whether rhythm categorization could already be present at birth as a product of nonlinearities of the nervous system. And to investigate this, we're recording EEG responses to rhythm in newborns during their first days of life, so to have minimal postnatal sensory motor experience. And this is a project that we're conducting in collaboration with Professor Sahar Moghimi and their lab in Amiens in the north of France. And so what we are doing, we are using an experimental design that is very similar to the one that we used in adults. And we have already collected some preliminary data, and preliminary results indicate that there is significant rhythm categorization already at birth. The categorical structure that we record from neural responses in newborns is, however, different from what we observe in neural and behavioral responses that we collected to the same rhythms in a group of adult participants. So this study is ongoing, but preliminary results suggest that some neurobiological mechanisms that are already present at birth may bias the neural representation of rhythm towards categorization, and rhythm categorization could then evolve through postnatal experience together with the development of neural circuitries. Then another project aims at investigating instead the effect of lifelong cultural experience on rhythm categorization. And to do so, our plan is to compare cultural groups with diverse musical training and experience in their lives. So we're starting to work on a project in which one group will consist of participants that are familiar with Western musical repertoire, while for the second group, we will test musicians from North Africa, and more specifically from a community from Morocco that is named the Gnawa. So the Gnawa musical repertoire is particularly interesting here for us because it contains rhythm categories that are not found in Western music. And what is very convenient for our cross-cultural study, the Gnawa community that is present in Brussels is among the biggest ones that are present outside Morocco, and is also very active in the musical practices in the city and in Belgium. And so this would allow us to conduct a cross-cultural study here in Brussels. And so the plan for the study will be to use sequences that are built from 3 interval rhythm patterns. And we created rhythm patterns in a temporal continuum from a 1:1:1 ratio to a 2:1:1 ratio. And these are rhythmic patterns that are that corresponds to universal rhythm categories that are found in Western music, but also all around the world. And interestingly, within this continuum of conditions, there is also the 4:3:3 rhythm ratio, which is prominent in the Gnawa musical repertoire, but is absent in Western music. So we had the opportunity to invite two Gnawa musicians in the lab, and we asked them to synchronize their tapping to the stimuli. And so I will show you some preliminary results here. So for the stimuli, as expected, we could again observe that as we move on this time continuum between the sequences are progressively more dissimilar from each other. Again, this is expected from the way that we constructed the stimuli. And when we look at the tapping responses, we could observe the two universal categories around the 1:1:1 and the 2:1:1 ratio. But importantly, we could also observe this time a third category, which corresponds to the culture-specific rhythm categories. And this was significant at the individual level. And so based on this, we're now planning to record both EEG and behavioral responses in a group of Gnawa musicians and in a control group of Western musicians to confirm this culture-driven plasticity. So with this, I would like to thank my colleagues and collaborators, as I'm very lucky to be working on this with a great team. So I would first like to thank my colleague Tomasz Lench, who is co-leading these projects with me, Professor Silvino Zaradan, the PI of the Rhythm and Brains Lab. And I would also like to thank our external collaborators, Professors Rainer Polak, Nori Jacobi, Manuel Varlet. Professor Sahar Mogimi is collaborating with us on the newborn project, while Helena Sechehe is collaborating with us on the Gnawa project. And I would like to thank you very much for your attention, and I'm happy to take questions and comments.
Thank you, Francesca, for this interesting talk and for sharing with us your results. And for the upcoming projects, they're super interesting, and we're looking forward to the final outcome. Let's proceed with the questions, if any. I will have a look at the participants list, and then we can go through the chat.
Yeah.
I can see a question popping up in the chat. Would you like me to read it for you, or you prefer to read it?
I can read it out loud. I mean, as you
Yes,
prefer.
yes, please go on.
Very interesting talk. Well, thank you. I apologize if I missed it, but what was the sample size for the categorization study in the first part of the talk? And where they screen for any current history of speech, language, or other neurodevelopmental disorder? Yeah, this is a very good question. No, I didn't mention it. So in the adult study, our sample size, we tested 18 participants. And when we recruit participants, we do ask them to not have any history of speech neurological or psychological disorder. So yes, we made sure about that.
Great, thank you. Right, I think it was crystal clear for everyone. If there are more questions, we can you can of course raise them later. Oh, there is another one. I'm sorry, we still have time for this one.
No, we didn't test this in, sorry, the question is if we tested it in school-age children. No, we did not, but it will be very interesting to actually be able to test this paradigm across different ages to see how rhythm categorization evolves through the lifespan. Yeah, that would be cool, but we didn't do that yet.
Great. Right, so if there are more questions, of course you can share them at the chat and we can read them at the end if there is time. There is another one. Right. How could you find the musical rhythms to test? Are there any backgrounds?
No, we actually used pure tones. So we used these sequences that were made by repeating rhythmic patterns that were made out of tones. So there wasn't any background. While instead, for the study that we are running in newborns, instead of pure tones, we used a conga sound, but just to be a little bit more infant friendly. And yeah, so there was no background.
Right. Let's proceed now with our second speaker. And as I said before, if there are more questions, I can read them at the end. So Francesca, stay with us. The second speaker of today is Carolyn Kroger, who is a third-year postdoctoral fellow working in the Cressy Hearing Research Institute at the University of Michigan. She holds a master's and a PhD in psychology and cognitive science from Michigan State University, working in the timing, attention, and perception lab. Her work investigates how people perceive timing and rhythm of auditory stimuli and how timing cues facilitate perception and action. So the title of today's talk is Influences of Pitch and Spatial Trajectories on Relative Timing Judgments in Auditory Sequences. Caroline, the floor is yours.
Sorry about that. I realized I was still muted. Thank you, Effie, for that introduction. And I'm excited to share some ongoing work in my postdoc here at University of Michigan. And so, yeah, let's get into it. So let's start off. Oh, let's see. There we go. Let's start by imagining a typical day when you're at a busy restaurant with your friend and you're having a conversation. And most of us do this fairly easily, but this is a simple act that's actually quite complex in terms of signal processing in the auditory system. So apart from listening to the voice of your friend, you also have constantly changing environmental sounds. For example, a phone ringing next to you, or dishes clanking, or other voices ongoing in the background, and they all combine into what we call an auditory scene that's processed by the auditory system. So the auditory system utilizes several cues to identify objects in the auditory scene. For example, the pitch or frequency of the voice, or the location of the sound source, and temporal cues such as sound onset. Simultaneous onset timing for different acoustic features is a strong cue for binding those features as part of an auditory object. For example, when someone says ah, the pitch, spatial location, and spectral content of that sound are bound by their shared onset, a cue for that individual voice. But in the context of a busy restaurant, the listener needs to not only bind simultaneous acoustic features that correspond to one word, they must also integrate these features over time to perceptually segregate different auditory objects, such as different talkers. In some cases, this feature integration causes perceptual biases, shown when physical changes in one feature influence the perception of a separate feature. And much research has been devoted to examining how individual features such as pitch, space, and timing independently affect auditory perception. However, these features do not operate in isolation, so the goal of this project is to understand how pitch, space, and timing cues interact to underpin our perception of auditory objects. So the auditory kappa effect is a perceptual illusion where pitch influences the perceived timing of tones in a sequence. Greater pitch separation leads participants to report greater temporal separation between tones. And the kappa effect has been demonstrated with an AXB paradigm. Each stimulus is a sequence of 3 tones where the A and B tones have fixed pitch and time separation. But the pitch and timing of the target X tone varies from trial to trial. And the target tone is always slightly early or slightly late relative to the temporal bisection point between A and B. So participants judge whether the target tone is closer in time to A or B by making a short long or long short response about the pattern of gaps between tones, explicitly ignoring pitch. So when target tone X is shifted earlier, the correct response is short long because X is closer in time to A. But when X is shifted later, the correct response is long short. So the kappa effect is represented by a bias in timing judgments based on task-irrelevant pitch information. When the pitch of X is shifted closer to the pitch of A, participants are more likely to report that X is closer in time to A, as shown by an increase in these short-long timing responses. Likewise, when the pitch of X is shifted closer to the pitch of B, participants are more likely to report that X is closer in time to B, as shown by an increase in long-short responses. So this illusion is a useful way to investigate feature interactions, and my talk today will demonstrate how we've used this illusion to examine the effects of pitch and space on perceived timing. So the current explanation for the kappa effect is based on the concept of auditory motion. The auditory motion hypothesis proposes that people perceive motion-like relations in auditory sequences. For example, one might hear a sequence of tones with increasing pitch frequency as moving upward, and that motion may be described as having a pitch velocity, which is total change in pitch over time. According to this hypothesis, the kappa effect arises due to violated expectations about pitch motion. When the pitch and timing of A and B are constant, people establish a stable velocity referent and, based on this pitch velocity, form expectations about when and where the target X should occur. When the target falls outside of the expected pitch trajectory, timing judgments are biased because larger pitch leaps are perceived as taking longer to traverse. So we propose an alternative hypothesis based on the concept of perceptual grouping. Perceptual grouping refers to the perception of separate stimuli as belonging together based on stimulus features. So this is like Gestalt grouping. And in this visual example, we may perceive the array of dots as 4 rows based on color similarity, or 4 columns based on proximity. And if one stimulus feature is ambiguous, you may rely on a more salient feature for perceptual grouping. According to the perceptual grouping hypothesis, when the timing between auditory tones is ambiguous, tones that are closer in pitch are grouped together, leading to the perception that they are closer together in time. I'll start off by showing results from 2 experiments testing these 2 competing hypotheses. The task was simple. After hearing a sequence of 3 sounds, participants report whether the pattern of gaps between sounds was short, long, or long, short by clicking on one of the 2 buttons shown here on the participant display. Experiment 1 was conducted just to replicate the kappa effect initially, and so we closely followed the original paradigm for this. Stimuli were these 3 tone sequences where A and B were always separated by 6 semitones and 728 milliseconds. The pitch and timing of the target tone X varied from trial to trial, where the target tone timing was either early or late with respect to the temporal midpoint between A and B. Timing offsets ranged from 4 to 16%, where negative timing offsets meant X was closer in time to A, and positive timing offsets were closer to B. The largest 16% timing offset creates a readily perceptible long-short or short-long pattern, but the smallest 4% timing offsets are very difficult to judge as the 2 time intervals are very close to being equal. Importantly, the pitch trajectory was always in the same direction, so sequential tones were always in ascending pitch order in this study, and I will note that this has been shown with tones going in descending order. So we were just trying to replicate this in the ascending pitch condition. So the target pitch was never below A or above B. And there were 5 target pitches. The 0 offset was exactly 3 semitones between A and B, or the target was shifted down 1 or 2 semitones closer to A, or up 1 or 2 semitones closer to B. So let's see if I can play a couple of examples to give you guys an idea of what this sounded like. Not sure if you guys could hear that very well, but in that example, that was short long, and that was shifted 16% early. So pretty clearly short gap than long gap. That one was long short. That was a 12% condition. So again, pretty clear that it was a long gap and then a short gap. And in that condition, it was 4% early, so a short, long one that was a lot harder to hear. So the data can be represented in this study as a psychrometric curve by displaying the proportion of long-short responses on the y-axis with the timing offset of the target tone on the x-axis. And I'm sure people here are very familiar with psychrometric functions, but I'll go through this just a little bit for anyone who is not as familiar. But 0 on the x-axis in this case would be the midpoint inter-onset interval between A and B, which was never actually presented in this study. But the dotted line shown would be perfect performance where participants always answered short long when the target timing was earlier and long short when the target was shifted later. And a bias in time perception is represented as a leftward or rightward shift in this theoretical curve. And we can quantify this bias with the point of subjective equality, or PSE. So PSE is the X value where the curve crosses 0.5 on the y-axis, or where participants responded long-short on 50% of trials. This is the theoretical point where target tone would need to be shifted in time to perceive two intervals as having equal duration, or two isochronous inter-onset intervals. The two durations are never actually equal, and people are always responding either short-long or long-short, but if there's no timing bias, the PSE should be 0. Here are results for the two largest target pitch offset conditions. So the red curve is when the target pitch was 2 semitones closer to B, and we see a leftward shift in the curve indicating an increase in long-short responses. The blue curve is when the target pitch was 2 semitones closer to A, and we see an opposite rightward shift in this curve indicating more short-long responses. So this is the kappa effect. The bias in perceived timing was stronger for the smaller timing offsets when timing was ambiguous. So when the timing is ambiguous, participants report that the target tone is closer in time to the tone that was closer in pitch. And we can see the shift in PSE where when the target pitch was 2 semitones closer to B, that corresponded to a target presented 20 milliseconds early with respect to isochrony. And similarly, PSE for when the pitch was closer to A corresponded to target presented 10 milliseconds late. And so here are the remaining conditions, and we see that the magnitude of timing bias scales with the shift in pitch. So in this experiment, we did successfully replicate the kappa effect, but in experiment 2, we wanted to test these competing auditory motion and perceptual grouping hypotheses by changing the pitch trajectory of each sequence. So just as in experiment 1, the pitch and timing of A and B were fixed, and all the target timing conditions were the same as in experiment 1. But this time we varied the pitch of the target tone in a different way. So unlike experiment 1, in experiment 2, the target pitch was always 1 or 2 semitones below A or above B. So the pitch trajectory changed on each trial. And all target pitches in this experiment introduced a change in the direction of pitch motion within a sequence. And these pitch trajectories changed direction from upward to downward on 50% of trials or from downward to upward on 50% of trials with randomized order. And so the random trajectory changes should prevent the formation of a stable velocity referent. So here are some examples of what these sequences would sound like. In that case, that's a long short sequence going down and back up. That was a short long sequence going up and back down. And another long short sequence. So the two competing hypotheses have opposite predictions for the results of this experiment. The auditory motion hypothesis predicts no kappa effect when pitch trajectories are unpredictable because there's no stable velocity referent for people to make predictions about when X should occur. But the perceptual grouping hypothesis predicts a kappa effect based on grouping the two tones that are closer together in pitch. And again, results are shown with these response curves for each target pitch condition. And so we do actually see a kappa effect for stimuli with these unpredictable pitch trajectories. On the left side, we still have a stimulus schematic showing two of those X conditions. And then on the right, we have our our psychometric functions for each of the 4 pitch conditions in this experiment. And there's a leftward shift in the red and pink curves when the target pitch was above the B pitch, and a rightward shift in the blue curves when the target pitch was below A. Thus, there was a bias where people reported that the target tone was closer in time to A when it was closer in pitch to A, or closer in time to B when it was closer in pitch to B. We also tested different pitch separations between tones A and B in this experiment. And here are stimulus schematics for 2 conditions showing 16 semitone and 18 semitone separations. We also tested 2 intermediate conditions with 8 and 14 semitones between A and B. And here are the results. We found that the kappa effect actually got larger with larger pitch leaps between tones. And this is maybe counterintuitive, but what we would predict based on the perceptual grouping hypothesis. This finding makes sense if we compare stimuli across conditions. So we saw that the largest kappa effect was for the condition with 18 semitones between A and B, but the larger pitch separations between A and B meant that the target was relatively closer in pitch to either A or B in those conditions. And we see that the bias for responding long-short when the target was higher than B can be explained by grouping the target B, and the bias for responding short-long when the target was lower than A can be explained by grouping the target pitch with A. However, in the 6 semitone condition, the pitch cue for grouping is much weaker because all pitches are relatively closer together. As an interim summary, experiment 1 replicated the kappa effect, and experiments 1 and 2 together provided initial support for perceptual grouping over auditory motion as an explanation for this effect. So regardless of the pitch motion, we found that participants grouped tone based on pitch proximity, leading to biased time perception. And thinking back to our original example of a busy restaurant, these results demonstrate that pitch and timing cues interact for grouping sounds as part of an auditory object. So if 2 people are talking at the same time, we can use the relative pitch and timing cues to perceptually segregate the 2 voices. And next, I wanted to test whether the kappa effect of pitch translates to auditory space. So we are motivated to test for a spatial kappa effect because this is the type of task that could have potential to be developed into a test of spatial hearing for people with hearing loss. So in experiment 3, we asked, how do changes in space affect relative timing judgments among sequential auditory events? In this experiment, we followed up on a previous set of studies to test for an auditory spatial kappa effect. And to our knowledge, this effect has not been reliably shown for 3-tone sequences or 3 sound sequences moving through auditory space. And the previous studies used simulated or virtual space using headphones and manipulating things like interaural time differences or interaural level differences. But we wanted to perform a stronger test of this effect by using real spatial manipulations. And additionally, the previous studies used pure tones when they did manipulate where they were, but we wanted to also use noise bursts that are easier to localize, as we know that pure tones are very difficult sounds to localize in space. So we adapted the classic kappa task from the pitch domain to the spatial domain. Participants sat in the center of a 360-degree speaker array and heard sequences of 3 brief noise bursts in an AXB paradigm, and each sound played from a different speaker location. Sounds A and B were fixed at 90 degrees apart, but the target sound X changed location from trial to trial. In some trials, the target sound was equally 45 degrees between A and B, and on other trials, X was shifted 15 to 30 degrees closer to A or closer to B, and sounds played sequentially from each speaker like so, A, X, and B. After hearing each sequence, participants reported whether the pattern of temporal gaps between tones was short-long or long-short, just as before, and they were instructed to ignore the location of sounds and only respond based on the timing. So here's an example of the noise bursts, but of course we can't manipulate the location of each over Zoom. So this is just to give you an idea. That would have been a long short pattern that they would have heard coming from 3 different speakers. So we tested normal hearing participants on this task, moving the sounds within 3/4 fields. So either 90 degrees in front or 90 degrees to the right or 90 degrees to the left. And we do see evidence for an auditory spatial kappa effect. So these results show that participants show a bias to respond that the target sound was closer in time to the sound that was closer in space. So in other words, when the target sound was shifted closer in space to the final sound, participants reported long-short timing pattern more and vice versa. So these are the results just from that front quarter field, that front 90-degree field. But we do see a similar effect across all three sound fields. So in conclusion, this study demonstrates how non-temporal auditory features can affect how we perceive onset timing of sequential auditory events. And we found that the kappa effect generalizes to the spatial domain. And overall, the results from all experiments support perceptual grouping. These experiments inform our understanding of feature binding within and across auditory events that underlies auditory object perception and stream segregation. Our ultimate goal is to adapt this task as a simple clinical test of auditory feature perception by examining the presence of timing biases based on task-irrelevant auditory features. So this timing illusion may be used to probe specific aspects of auditory perception that are affected by hearing loss. So we know that hearing impairments cause problems with understanding speech and noise, and we're currently testing for auditory spatial kappa effects in individuals with single-sided deafness to assess the extent to which this is caused by problems with auditory grouping based on sound source localization in this population. We're also conducting an experiment with both pitch and spatial cues that congruently or incongruently promote grouping to assess how aging affects auditory feature binding in these different feature domains. And we're also moving forward with examining effects of higher level features such as musical harmonies and speech, and to see if there are timing biases in or based on kind of higher level feature manipulations and grouping in that sense. So with that, I would like to thank my PI, Dr. Mehta, as well as our wonderful collaborators and research assistants, and of course our funding sources. And thank you all for listening, and I'll take any questions.
Thank you, Caroline, for your talk. I had a question, but you shortly answered that. I was about to ask if you're going to combine speech and space, but I'm looking forward to your results. Let's see if there are questions for you.
I don't know that I can see chat from here. I do see a hand, Martin.
Yes.
Yeah, thank you
Dr.
very much.
Martin Weimer.
Yeah, thank you for the talk. I find the hypothesis really plausible, and it's quite convincing, your results. I wonder, in your second experiment, whether if you assume still a motion or a motion from the low pitch to the very high pitch and then back to that other pitch, whether that should not also then increase when you increase the pitch differences. You know what I mean? When you interpret the high differences in pitch as longer time, shouldn't you then have exactly the same or similar effects?
There is a potential for that kind of effect. The problem, I guess, is then needing to update that type of model to account for these differences in trajectory, because it's operating on this assumption of constant velocity. So going back to the original model, which was developed by Jones and Wang in 1982, that was applied to visual motion. And then they adapted it in a paper in 2016, I think, to be in the auditory domain. So first, I think you'd have to kind of reconcile that velocity is not constant, but you could think of a version where you'd have a parameter that allows for changes in trajectory. So the next thing that we're doing with this is actually testing models, behavioral phenomenological models of each of these hypotheses to try and tease that apart. But that's a great question.
Great, thanks. Maybe one more obvious question. So what about kappa? What about tau?
Yeah, so we haven't done anything testing tau. It's been tested extensively. Obviously, I'm more focused on the time perception aspect, so I'm more curious about how other features affect timing. But for those who are not familiar, the tau effect is the reverse, where the timing, the onsets actually affect things like the perception of pitch or location. So yeah, we haven't tested that yet, but it's certainly still on the table. And I think that any model needs to be able to similarly account for both.
Great, thank you.
Yeah, thank you. Good question. Sorry. Oops.
Yes, we do have time for a couple of more questions, if any. Let me go through the chat too.
Yes, I don't think I'm seeing chat from my screen. So if there are any questions in chat, feel free to shout those out.
I will read them out loud for you, but I think they're still for the previous talk. We still have questions or a discussion going on there. Right. So if there are no more questions for Caroline, we will close this meeting earlier, but feel free if you come up with questions to contact our speakers or us, and we can forward the answer. I would like to thank our speakers for delivering these talks today. They were super interesting, and we're looking forward to your future work. So thank you all for attending, and we will see you again in a month. Please stay tuned because this talk will be uploaded on YouTube channel, so you can asynchronously watch it or share it with your colleagues. I'll see you in 1 month from now. See you soon. Bye.
Bye. Thank you.